So I'd like to begin the session, and I have a pleasure to begin the discussion on biosimilars, regulatory framework and quality consideration, and also the scientific and clinical requirements around development of biosimilars. And I would like briefly just to say that what is great about this session, we have speakers representing different stakeholders, which will be regulatory agency. We have originator companies presenting their approach and their views on how these regulations should shape up in Russia and globally. And also we have biosimilar companies developing biosimilar products. So I think that hopefully will allow us to achieve the following scope of the meeting. By the end of this three presentation, which I would like to ensure that each speaker adheres to approximately 20 minutes of the presentation, and then at 4 o'clock we will begin Q&A session, and please sum up some of your questions. I think the main goal is to try to come up with some consensus, or at least with some principles which we would like to take forward, what lessons we should take forward with biosimilars, and what we need to look in the future.
So, good afternoon, ladies and gentlemen. I think that the issues related to regulation and assessment of biosimilar quality and their international practice is a very, very interesting topic that we're going to discuss today. So, in what way should we regulate this process? How do we bring together data and findings both abroad and in Russian Federation? I think that this is something that we're going to discuss in this session. So without further ado, I'd like to give you the first presentation, which is dedicated to specifics of regulatory assessment of biosimilar products in the European Union, assessment of preclinical and clinical data by Alexey Kudrin. Sir, you have the floor.
I would like to focus in my presentation, first of all, on the fact how successful some of the biosimilar products actually becoming. So, first of all, biosimilar actually already been approved in multiple regions, and we have 14 biosimilar products approved in the European Union. But actually, market overall is still remain quite modest. If you look at these figures from IMS from 2012, and forecasts suggest that the uptake of biosimilars will be remaining quite slow compared to the overall biologic market for the reasons of substitution issues, and also for the reasons that regulatory requirements remain sometimes quite difficult for companies to overcome. However, lessons learned from European markets, however, have been quite positive so far. So we have approximately 60% of erythropoietin market occupied by biosimilar erythropoietins. For colonial-stimulating hormone factors, filgrastims, 20%, and for growth hormones, 20%. We also know that the uptake is uneven between different countries and depends on different factors. So, for example, for growth hormone, the very much factor which determines switching from originator to biosimilar is dependent on pediatric endocrinologists. We're talking about small children receiving these hormones for years. So there is a sort of tendency of prescribers not to switch patients because they are scared of potential consequences. Whilst with filgrastims, where cyclical administration is often used, it's a very common phenomenon to switch from originator to the biosimilar product. What is also interesting that pricing pressures arising from the number of products coming. So, for example, with erythropoietin, originator managed to drop discount to almost 82%, which significantly makes difficult for biosimilar erythropoietin products to come on the market and trying to compete. So you can see that there is a huge dynamics of these products coming through. And this is figures representing how different uptake of biosimilars across 10 countries. And as you can see, for example, in Germany, you can see an increase of uptake, and certainly some countries like Romania and for example, Sweden and Norway, they now are taking quite significant bulk of biosimilar products. But some other countries seems to be lagging behind, and there are various factors which influence this process. The experience overall with EMA, and I'm proud to say that we managed to be one of the first agencies in Europe, worldwide, to actually drive this legislation forward, and already now take lessons out of this, how this products working in hundreds of thousands of patients. So 14 biosimilars been approved, and the history now span into 2006, so it's a long history already, and more than 40 scientific advice procedures for biosimilar monoclonal antibodies and other classes of products been received by EMA in the last few years. What lessons we learned with regulatory review of biosimilar monoclonal antibodies, and this is a slide from Christian Schneider, who is the head of Biosimilar Working Party. In general, this dossier is often accompanied with major issues, and companies have to address quite a lot of quality, non-clinical and clinical issues. Some of those called major objections, which constitute an issue which potentially might preclude for this product to actually get a license. However, those issues are also resolvable, so a company can find a way to justify the approach forward. And whilst this issue has been raised, companies who managed to get license managed to kind of get this major objection sorted. The length of biosimilar review process is slightly longer than with originator products, because that's inevitably the case, because the rapporteurs of EMA have to look sometimes in originator dossier to make comparability exercise on regulatory level and ensure that the data presented by the biosimilar company match into what originator company submitted in first place. So it takes longer process to do scientific evaluation of biosimilars. We had a number of biosimilar targets in the past, from filgrastims to Enbrel, but now the new class of biosimilars come in as monoclonal antibodies. And I can only say, because it's already available in public domain, that biosimilar infliximab is currently under assessment, and there are final stages of procedure which you will hear about outcome in the next few months. And certainly there are a number of biosimilar monoclonal antibodies which are currently in scientific advice stage, already in phase 3 ongoing studies, like biosimilar trastuzumabs, and there are apparently about 40 biosimilar rituximabs in clinical stage of development. I would like to summarize definition, which is very important because there is a lot of confusion about terminology of biosimilars, and in Europe we're now constantly talking about it. There are products which shouldn't be called biosimilars because they're not been developed as biosimilars. Similar biological medicinal product is a new product claimed to be similar to a reference medicinal product, and it doesn't meet the definition of generic. So the reference from current overarching guideline point of view, and new guideline is currently under revision, is that reference should be approved in Europe, or at least in one of the European countries. And biosimilarity development paths should be not designed in opportunistic fashion, but has to be from the start. And it's very different to what some companies think. Oh, we've been developing a new product, but now we think it might be biosimilar. No, it has to be original theory, original principle to develop biosimilar products. So everything in this pathway has to be adjusted towards that. The quality target profile should be achieved in terms of representing or mimicking glycosylation profile of the originator, and also resembling the presence of impurities. It's very important that as authorized biosimilars generally administered via the same route of administration and with the same posology. And we also insist that primary structure should be identical to the reference biological product. So we also agree that biosimilar pathway is not trying to reinvent wheel in terms of proving benefits, clinical safety and efficacy. It's biosimilarity what need to be proven. And major important argument we have is that biosimilarity life cycle will be different from reference product, because inevitably reference product also changing. And some companies which hold reference product actually deliberately change this product in order to defend their product from biosimilar arrivals. So it's inevitable that pathway will be different, and there is no need to constantly follow through post-marketing phase that biosimilar will remain the same as reference product. There will be some differences. We have to say that there are some of these so-called biobetters, which the terminology confusingly used by a number of different stakeholders. And I strongly advise companies to think about before they call something biobetters. It's been a mixed bag concept, which originally came from Dr. Reddy, and now biobetters are sort of called either type of biologics with new features, or something might have different glycosylation features which might be better, but we actually haven't seen any cases so far. They may have completely different and atypical target product profile, different exaggregated PK properties, and different efficacy and safety features. So we usually recommend for those companies to go through new active substance route rather than try to attain biosimilarity path. And the same FDA shares the same view. The biosimilar concept is a stepwise approach, and one needs to go through those steps. And if you jump through several steps at the same time, you embark on a risk that once you started clinical trials, bioquality, comparability not finished, you're actually conducting a useless trial. However, comparability tools vary in terms of their sensitivity. If you start from quality analytical characterization, they are very sensitive tools. Sometimes they might actually unravel changes which may not be relevant on clinical grounds. However, clinical studies, quite crude way of looking at the differences, and for that reason we insist that those studies need to be done in appropriate patient population. But basically, essentially what we insist on that pyramid, which you already seen today on one of the slides. During review of biosimilar submission, it's a holistic review where assessors on quality, non-clinical and clinical and pharmacovigilance interact together, because biosimilarity is a totality of the evidence which matters. And that's what actually creates the final view on whether it's actually biosimilar or not. And this is very different to other biotherapeutic products. We do interact when we assess a new biologic, but with biosimilars it's particularly important because any data from in vitro assays might translate to clinical differences. And once everybody is on the same page, decision can be made very swiftly. As I mentioned, we have a lot of guidelines, and I don't want to go one by one, but these annexes cover quite a range of different classes of products, from insulins to low molecular weight heparins, and of course the monoclonal antibody guideline, which was introduced last year as one of the key one covering monoclonal antibodies. A little bit of potential difference which we encounter in terms of looking at the FDA approach and EMA approach. FDA, I think, looking to attain highly similar pattern, whilst with EMA we expect that there will be similar, but there might be more differences potentially. I think one of the key differences FDA demands from companies at this moment is that PK study as initial comparability exercise in clinical trial is done using reference product issued available in US, with reference product in Europe, and also biosimilar. So 3-arm trial. Whilst in Europe, we actually waive this requirement in terms of asking companies to demonstrate comparability on a quality level between US and EU available reference products. I would not touch non-clinical data to extent because I have my colleague, Dr. Haim, who will cover all the non-clinical requirements in details. But essentially, there is a stepwise approach with non-clinical development. And then once we came to the conclusion that there are sufficiently similar profile with non-clinical data, the clinical comparability exercise is also stepwise. So PK and PD data is a paramount feature of biosimilarity, and it should be demonstrated in all cases. Then there is also a necessity to conduct efficacy trial, which is usually equivalence trial, but not bioequivalence study. So there is a confusion between bioequivalence and therapeutic equivalence. That's not the same thing. So the requirements for the PK study is to look at the absorption and elimination of the monoclonal antibody, and choice of design and equivalence margins should be justified. But we prefer the usual one, 80 to 125% for Cmax. And then pharmacodynamics should be looked at either as a part of efficacy study or as a part of the PK-PD study. And in fact, sometimes it's possible to integrate everything in one pivotal study, and some companies do so. This is an example of PK or PD failure, when you can see on the left with PK-soluble insulin there are differences between test and reference product. And similarly on the right you can see difference in terms of the level of glycemic control, and again the products definitely not overlapping. So in this case we would be expecting a very similar pattern. An example of PK and PD success when the area under the curve for the product actually surely falls within 80 to 125 and actually falls within the sort of median range. So it's right in the middle of this sort of interval. So that's success to us. PK studies in healthy volunteers is often the first step when it's done with biosimilar monoclonal antibody. And actually there are ethical challenges of doing such studies. So for example, I mentioned today about natalizumab with PML. Of course, it's going to be very tricky to justify healthy volunteers to receive such product. But for one dose, single dose infliximab or biosimilar adalimumab, it is possible. And in fact, we've seen proposals of doing single dose or bevacizumab in the healthy normal volunteers. What is ideal about the single dose study? That it generates information about, first of all, about variability and greater sensitivity of PK data in immunocompetent patient healthy volunteers rather than in patients. And certainly it also allows to see, if you look at the 5 half-lives, whether there are any changes at the terminal stage or terminal phase of elimination. So that's the key part of this study. So if all of that's similar within 80 to 125, company can proceed with next stage. For some products like Enbrel or etanercept, it's possible to do crossover studies because half-life is very short. It's usually around 2 to 3 days. And certainly, if not possible to conduct study in volunteers, we normally expect patients' data or multiple dose PK and PD at steady level in patients, and that can be done as a part of equivalence trial. Sampling times can be justified, and it has to be justified. And what a general recommendation that sampling times should be sufficient around predicted Cmax of the biosimilar or originator product. And in PKs, in CD studies, the primary endpoint should be AUC from 0 to indefinite time, and secondary endpoints are the PK parameters. And for MD studies, it's truncated AUC. From statistical tests, as I mentioned, the margins should be around this level. In some cases, widening might be justified, but usually we do not welcome that. And then once all of that been described, adequately powered randomized double-blind parallel group design study is usually preceded. And in this study, equivalence margin should be justified. And for example, for biosimilar adalimumab or infliximab, it should be equivalence of either ACR20 or DAS28, which are sort of rheumatoid arthritis score systems, which allow us to see whether actually the efficacy is very similar. And generally, efficacy and safety studies should be done with one route, or if there are several routes of administration approved, then there might be a little bridging study required. The patient population should be homogeneous and sensitive enough, what means that ideally it should be a patient population receiving, for example, as lesser immunosuppression therapies or maybe one combination to minimize different heterogeneity. And certainly from the dose selection point of view, one can choose something which is in a steep dose finding curve and something which is very sensitive to identify potential differences. How to justify the margin? Well, first of all, companies are encouraged to look at the literature, public reports for the originator company, and if, for example, effect size was around 42%, and placebo was given in those studies 18%, the difference between that would be 24%, and the margin for equivalence will be half of that. So that's generally what we expect. We expect margin size for efficacy trials justified, of course, at the level of not greater than 15%. So in some cases, greater percentages allowed, but certainly that sort of level we expect. I have to say that there are now biosimilar monoclonal antibodies for oncology, and for those, the dilemma is that studies to look at the overall response and progression-free survival from equivalence perspective create a very large sample size. And certainly there we're looking now at the further ways forward in terms of looking at pharmacodynamic readouts and pharmacodynamic equivalence trials. I will not cover immunogenicity, but one thing here is in general we expect data at 12 months presented. There are some exceptions, like with, for example, etanercept, but generally discussions around 12 months data should be presented because that's meaningful amount of time for patients to mount immune response and look at the potential differences. And extrapolation of indication is one of the serious challenges for companies to overcome in biosimilar submissions. If the mode of or if the mode of action or mechanism of action is the same, then it's more straightforward. But if there are differences, like with rituximab, it's very difficult because there is autoimmune indication and then there is an oncology indication. So in this case, we support position that rheumatoid arthritis is the key comparability indication, and that supportive data, some supportive trial should be generated in lymphoma or oncology indication. And the outcomes of extrapolation exercise is based on totality of biosimilar. Actually, there are two schools of thought within EMA, so it's not fully been sort of solidified yet. There are groups of experts who think that extrapolation might be dependent sort of on totality of evidence, and it's either everything, all indications, or nothing. And there are some groups of experts think that there might be partial indications granted, and through additional supportive study, additional indications might enter into the label. So we will see how these things will shape up in the years to come. Pharmacovigilance of biosimilars is important, as we mentioned today. Pre-approval databases will be rarely exceeding 500 to 700 patients. So the emphasis will be on risk management plan and looking at the safety data gathering through the post-marketing phase. Some of the observational studies, for example, and the way how companies collect data in extrapolated indications will be especially important. So because we simply will not have any clinical data at the time of approval in extrapolated indications. So risk management plan is very well adjusted for that. And certainly from the immunogenicity, something we do not actually regulate in Europe at the European Medicines Agency point of view, interchangeability and substitution, but national agencies do. They have within the legal remit to look at the switching data and extrapolation or on interchangeability data. And so that's a very contentious issue which sort of I'd like to cover in the end. From the product labeling point of view, generally the position that mostly biosimilar will have the same label as originator company, like with generic products in a way, but some additional wording will be allowed to describe, for example, results of the study which was used to support that equivalence trial to support the claim for biosimilarity. And might be some specific statements in pharmacodynamic properties. But this is again very evolving concept, and this may change slightly with different type of biosimilar monoclonal antibodies coming through. What is important from WHO perspective that there are different level of interchangeability. So there is interchangeability where it's driven by prescribers with agreement with prescriber. There is substitution which requires some sort of pharmacy intervention but without consultation with medical practitioner. And there is automated or automatic substitution which is dictated by policyholders and governments. So situation in Europe is very diverse in that sense. There is now sort of mixture of automatic substitution in some countries because of the budgetary constraints for those approved biosimilars which I mentioned. And certainly there is some sort of elements of substitutability. The way how it's controlled now from the point of view of EMA, we have no expectation for pre-approval switching data, but we're looking through pharmacovigilance data coming through. With FDA, there is no precise study designs to address this, but we've seen already proposals how to address this in pivotal biosimilar studies. They do, for example, switch from the originator, half of the arm of the originator product, switch to biosimilars through 6 months, and half will remain an originator, and then they compare what happens with immunogenicity. And we accepted this proposal as long as it doesn't affect the power of entire study to address immunogenicity. So from the point of view of EMA, we and MHRA specifically for UK market, we look at the importance of how biologics prescribed and how biosimilars named by the product name, brand name, and also by the batch name, batch number. So we can then trace any safety to specific biosimilar product. And so there are very important warnings for some of the products in terms of traceability. So prescribers are warned about what is actually needed in terms of tracing products forward. And I mentioned already on this slide in the previous presentation that biosimilars constitute product under surveillance or intensive surveillance in the first few years after marketing. So I think then I will finish and I leave floor to the next presentation. Thank you.
Good afternoon. First, I want to thank the organizers for inviting me for this presentation. And I want to stress the fact that the opinions and conclusions presented on the slides are my personal position, and not necessarily the position of any official regulatory agency. First, I want to make some remarks why we have a specific legislation for biosimilars in the EU, and then I want to focus on the non-clinical aspects. Now, the current definition for a biosimilar in the EU is that a biosimilar is a biological medicinal product that contains a version of the active substance of an already authorized reference medicinal product. And based on a comprehensive comparability exercise, a biosimilar demonstrates similarity in terms of quality, biological activity, safety, and efficacy. Now, obviously, there are differences between biosimilars and chemical generics. These relate to the size of the products. Furthermore, glycosylation may introduce further diversification in the case of the biosimilars. So that for the biosimilars, for the glycosylated forms at least, we can state that the products are a mixture of closely related active substances. Now, glycosylation will also influence protein charge, folding, and stability, will have influence on binding affinity, also affect immunogenicity, and of importance, also pharmacokinetics, inclusive tissue distribution. Now, also important for the biosimilars is the fact that they are produced on basis of a complex manufacturing process. Now, even minor changes in this process may introduce significant changes which may have consequences for post-translational modification, impurity profile, and also immunogenicity. So the question was, can a generic approach be used for marketing approval of biosimilars? Just to remind, generic means the same qualitative and quantitative composition, same pharmaceutical form and bioequivalence with reference product. Now, as we have seen, the answer is no, due to complexity of biological biotechnology-derived medicinal products. The generic approach is not appropriate for these biosimilars, and to establish safety and efficacy, the so-called biosimilar approach, this means a comprehensive comparability exercise is needed. The basis for this approach is laid down in the Directive 2001/83/EC in Article 10.4, which makes reference to the annex, and this annex makes reference to the guideline on similar biological medicinal products, which contains the main references to more specific points. So, based on this guideline on similar biological medicinal products, a framework for non-clinical evaluation of biosimilars has been developed in the EU, consisting on an overarching guideline for protein-derived products and some product-specific guidelines, and also on a product-specific guideline for non-protein product for the low molecular weight heparins. Now, as you can see, the first guidelines were finalized in 2005 and have been expanded to specific issues then. Now we have already the second generation of non-clinical guidelines. Some guidelines have been finalized already, and some are in revision currently. As I have already mentioned, to establish whether biosimilar demonstrates similarity to a reference medicinal products, quality characteristics, biological activity, safety, and efficacy have to be proven in a so-called comparability exercise. This exercise has several components, normally a very extensive quality part, then non-clinical studies, which may comprise in vitro studies and in vivo studies, and finally the clinical studies. Now, this slide shows that for the quality, the efforts taken will normally even exceed those that the originator has taken, because you have to characterize your product and, in addition, also the reference product. Now, for the non-clinical and the clinical, normally the amount of studies is reduced, and the biosimilar makes reference to the data produced by the originator. Finally, a decision has to be made whether the data produced for the biosimilar in these different parts of the comparability exercise are sufficient to claim biosimilarity overall. Now, for the non-clinical evaluation, it is important that the studies should be performed before initiating clinical trials. And furthermore, and this is quite new, a stepwise approach should be applied, which consists in the step 1, in vitro studies. When at step 2, a decision whether any additional in vivo studies will be necessary, and only if needed, step 3, in vivo animal studies. Just to show that this approach has changed from the first generation guidelines, the initial guidelines had this linear approach, which requested non-clinical in vitro studies and obligatory in vivo studies. Now the approach taken by the second generation guidelines is to perform in vitro studies and then make an evaluation and a decision whether in vivo animal data will be needed or whether clinical studies can be started at once. Yes, the in vitro studies, we should cover the spectrum of pharmacological toxicology aspects known from a reference medicinal product. And the aim is to assess any differences in biological activity between the biosimilar and reference medicine product. And to do this, data from a number of comparative in vitro studies are usually required. Such studies could include, for example, binding to targets, and on a functional level, signal transduction, functional activity, and viability of cells involved, and known to be of relevance from the pharmacological effects of a reference product. These in vitro studies should be comparative in nature, that's very important, and should be designed not just to assess the response per se, but to detect differences between the biosimilar and the reference product, if such differences exist. The parameters should be sensitive enough to detect the differences, and if possible, a concentration activity or binding activity relationship between the biosimilar and the reference product, covering a concentration range which might show differences, if applied, should be used. Furthermore, an appropriate number of batches should be used for comparative exercise. Since these in vitro assays are often more specific and sensitive to detect differences between the biosimilar and the reference product, they are considered as paramount for the non-clinical evaluation. However, the applicant should justify that the used assays are predictive for the in vivo situation. If these in vitro studies and the quality assays do indicate significant differences between the intended biosimilar and the reference product, which make it unlikely that the biosimilar will eventually be established, a standalone development should be considered instead. Now we come to the step 2, determination of the need for in vivo studies. Generally spoken, biotechnology-derived proteins may mediate in vivo effects that cannot be fully elucidated by in vitro studies. I'll bring a few examples right on. Therefore, in specific cases, non-clinical in vivo studies may be necessary to provide complementary information, provided that a relevant in vivo model with regard to species or design is available. So factors to be considered when the need for in vivo non-clinical studies is evaluated include, but are not restricted to, quality factors and to pharmacokinetic factors. Quality-related arguments could be the presence of relevant quality attributes that have not been detected in the reference product, or significant quantitative difference in quality attributes, or relevant differences in formulation. Although each of these factors may not necessarily warrant in vivo testing, these issues should be considered together to assess the level of concern and whether there is a need for in vivo testing. Furthermore, if product inherent factors that impact PK and or biodistribution, like for example extensive glycolation, cannot sufficiently be characterized on a quality and in vitro level, in vivo studies may be necessary. The applicant should then carefully consider whether this assay should be performed in animals or as part of clinical testing, e.g. in healthy volunteers. Now we come to the decision process at step 2. If a comparability exercise in vitro studies and the quality assays are considered satisfactory and no factors of concern are identified in step 2, or these factors do not block giant entrance into humans, then an in vivo animal study may not be considered necessary. Now, if there is a need for additional in vivo information, the availability of a relevant animal species or of another relevant model, like a transgenic animal or a transplant model, should be considered. If such a model is not available, the applicant may choose to proceed to human studies directly, taking into account principles to mitigate any potential risk, like for example described in ICH guideline M3. Now we come to step 3. We suppose now that an in vivo study is necessary. When the spectrum of studies which is deemed necessary depends on the need for additional information. So dependent on the outcome of step 1 and 2, either a PK and or PD and or safety study will then be required. Studies regarding safety pharmacology, reproduction toxicology, and carcinogenicity are not required for non-clinical testing of a biosimilar. Now, general problems with in vivo evaluation of biosimilars in animals are the following. These recombinant human proteins may show species-specific pharmacodynamic activity, and it may be difficult to identify relevant species other than non-human primates. And as foreign proteins, they will usually elicit an antibody response in long-term animal studies, and this means long-term studies, let's say longer than 2 bis 4 weeks, may result in the formation of antibody complexes, and when the interpretation of such studies will be difficult. Now, animal studies should be designed to maximize the information obtained. The principle of the 3 R's, this means replacement, refinement, reduction, should be considered when designing any in vivo animal study. And depending on the endpoints needed, it may not be necessary to sacrifice animals at the end of the study. The regulatory basis for this application of the 3R principles is laid down in Directive 2010/63 EC on the protection of animals used for scientific purposes. Now, let's say we have PK/PD studies to design when the model allows the pharmacokinetics and the pharmacodynamics of a biosimilar and the reference product should be quantitatively compared, including concentration response assessment covering the therapeutic range in humans. The duration of a study should be justified taking into consideration the PK behavior of a biosimilar and its intended clinical use. For safety studies, a flexible approach should be considered, in particular if non-human primates are the only relevant species. In general, the conduct of standard repeat dose toxicity studies in non-human primates is not recommended. If appropriately justified, a repeated dose toxicity study with refined design, for example, using just 1 dose level and or just 1 gender and no recovery animals, or an in-life evaluation of safety parameters such as clinical signs, body weight, or vital functions may be considered. Studies in local tolerance are usually not required. However, if excipients are introduced for which there is no or little experience, then local tolerance may need to be evaluated. In such a case, if there are other in vivo studies, local tolerance may be included in the design of these studies. Now, the conduct of toxicity studies in non-relevant species, let's say to assess unspecific toxicity related to impurities, is not recommended. Due to different production processes between the biosimilar and the reference product, qualitative differences of process-related impurities will occur. Now the aim of the quality production of this product is to keep the level of such impurities as low as possible. So this is the best strategy to minimize risk associated with impurities. Concerning the immunogenic potential, qualitative or quantitative differences of product-related variants, for example, related to glycosylation or charge, may have an effect on immunogenic potential and the potential to cause hypersensitivity. However, these effects are difficult to predict from animal studies and should be further assessed in clinical studies. Nevertheless, immunogenicity assessment may be necessary for interpretation of in vivo studies in animals because antibody formation may influence pharmacokinetics. Therefore, blood samples should be taken and stored for future evaluation if they're needed. Now I want to present just a few examples of recently finalized product-specific guidelines for the biosimilars. For the monoclonal antibodies, the step 1 program looks as following. There should be assays on binding to target antigens, Fc receptors and complement, and also functional activity investigated with regard to FAB-associated functions, Fc-associated functions. Now, ADCC and CDC may be only important for products which target cell-mediated events, not for soluble targets. Perhaps one step back. If there are no specific concerns in step 1 and quality assay also does not raise specific concerns, then for the monoclonals, proceeding to clinical evaluation may be possible directly. Now for the interferons in step 1, for the in vitro studies, there is also an assay package including receptor binding studies and functional assays like antiviral effects, antiproliferative effects, and immunomodulatory effects. Also here, if there are no specific concerns from this in vitro studies and quality assays are okay, then a direct continuation with clinical studies may be possible. As a last example, for follicle-stimulating hormone, also in step 1, we have receptor binding studies and receptor activation studies. However, in this case, step 2 may lead to the following conclusion. Because follicle-stimulating hormone is a highly glycosylated protein, in vitro studies may not fully reflect the more complex situation in vivo. Hence, the need for additional comparative in vivo animal studies should be considered in this case. And actually, normally, the potency biological activity of FSA is today established in the Steelman-Pooley assay, which measures the FSA-induced increases in ovarian weight in female rats. Now, the applicant may choose to use a different bioassay, maybe in vivo, ex vivo, in vitro. However, in this case, he has to justify this approach and present data that this approach is really comparable to the established Steelman-Pooley assay. So that's all from my point. I thank you for your attention.
So I would like to begin my report with the definition of what evolution is. If you refer to the Soviet encyclopedia, then the evolution as a system involves a long-term changes of preceding states. What does it mean? It actually means that the evolution with regards to biosimilar products in Russia has been taking a longer route compared to that of other countries. But that said, this process is on the way. It doesn't stop. So I'll give you highlights of these proceedings. Now, what are the prerequisites to regulating approaches towards assessment of biosimilar products? And the thing is that these biosimilar products are quite young. They appeared in the market not so long ago, but their appearance raised a lot of issues. How do you control these? What's their efficacy? What's the safety of these products? But the thing is that these questions were asked and referred to not only towards the manufacturers, but towards the patients as well. Can these be substitutes? What about interchangeability? So the situation with regards to biotherapeutical products, and I believe that biosimilar, bioanalog, and biotherapeutical products are pretty much the same kind of notions. So anyways, of course, having a biosimilar on the market after the expiry of the patent of a original drug is a natural process from the viewpoint of economic standpoint, but at the same time, the patient is supposed to be provided with the most efficient and the most safe medicines out there. So, control the approach towards biosimilars. Let me talk about that. Where do we see it? We believe that there are several key participants, stakeholders. First stakeholder is the Ministry of Healthcare. And the Ministry of Healthcare has to have two goals in front of it. First one is economic efficiency, and the second one is therapeutical efficiency of these biosimilar products. Now, the second stakeholder is a manufacturer, and they have their own agenda. And their agenda is to, as a commercial company, of course, to put these products on the market as quickly as possible, and to make sure that there is a reliable distribution of these products. Now, the third stakeholder is the patients community, which want most rigid requirements from the regulatory bodies, and of course they want efficacy of these groups of medicines. Now, and for this audience, I would like to present you with a new term. We've developed this issue and decided to come up with this term called regulatory inertia. And I'm speaking specifically about biosimilar products. So how come? Where did that come from? So let me explain. The thing is that the first regulation with regards to biosimilar products happened in the United States and Europe, and the first guiding principles came up is in 85, 87. So the expiration of a patent in the 2004 with regards to insulin and maltodextrin, as well as erythropoietin. So that happened in 2004, and the first guiding principle by EMA was only in 2005. So what about the situation for WHO? The same situation there. So you know that there is a multiple reviewing sessions by both EMA and FDA. They all reviewing their practices and generations of guidelines. Now, what about the Russian Federation? What do we consider a biological medical product? So this is the following group of products. These are immunological medical products, products derived from donors' blood and plasma. These are also medicines obtained through the so-called biotechnologies, and these are the products of advanced therapy. These are somatic cells, gene therapy, as well as tissue therapy products. Now, this slide shows the evolutionary track. This is the evolution regulatory pathway. So this is starting from 1994. Of course, it doesn't date back to 1994, but we started to take it from that year. So this is the regulatory pathway evolution from that time until today. And this is specifically for biosimilar products. I would like to draw your attention to the fact that before 1998, which is a very important year because this is where the Federal Act was enacted on medical products. We had basically two standalone procedures: a standalone procedure with regards to immunobiological products and another one with regards to ready medical medicines. Now, but in 1998 they tried to combine these two procedures, but these were two standalone procedures that would be overlapping, and there were a lot of differences and clashes of opinion. So merging these two ideas, these two subjects, the two regulators such as Rospotrебnadzor and Roszdravnadzor, and also their charters and regulations, they didn't happen basically until 2010. So what did happen in terms of regulation of immunobiological medicines in 2010? Well, 2010, the requirements were finally merged towards immunobiological products and medicines. So these requirements became one. They were unified, and all these misunderstanding was completely removed from the market with regards to these groups of medicines. Now, I would like to also give you an idea of the way immunobiological medicine as a notion in Russia was formulated. It was 1992, 1994, then also 1998. There are two laws. First one is about immunoprophylactics and the other one on medical products. These are two federal acts which give the list of these medicines and also define the goals. And in 2010, a new legislation comes around with a new terminology, the new notion of immunobiological medicine, the new definition, focusing on their origin and also showing the goal, which is, of course, diagnostics, prophylactics, and treatment. So let's talk about today. What do we have basically in Russia at the moment? We're discussing this issue, and I think that quite soon there will be a brand new legislation, and this would finally fix the definition of what a biosimilar medical product actually is. Now let me express the expert opinion viewpoint. This is our expert organization, so this is what we believe it is. These are biological medical products which were developed using gene engineering or biotechnology methods. It's similar to reference product, and by reference product we believe an original product in terms of quality, safety and efficacy, and the differences identified have no therapeutical significance and don't fall in the definition of a generic medical product. And I would like to draw the attention of the audience on the following notion. On numerous occasions, we've attempted to translate biosimilar as a term into Russian. And the thing is that from a scientific standpoint and expert standpoint, we believe that these two terms, biosimilar and bioanalog, are synonymous. It's actually the same thing. So what happened in Russia after the enactment of Law 61, which is a federal law? Well, for the first time ever, the requirements to conduct of expertise were set forth, as well as requirements to peer reviews. Now, after we've set up a pan-Russian expertise center, we've developed a unified requirement. So what is the current expert practice? Or peer review practice, as we call it. Well, at the moment, we don't have any regulatory acts that control biosimilars specifically. So it might seem that during the peer review and experts they treat biosimilars as if they treat reproduced medical products or generics. It's not so, believe me, until 2010. And this has to do with the fact that there were two regulators basically with different structures and different regulations. Only to that, believe me. And so the review and the analysis of biosimilar products and medicines would often lead to very contradictory requirements. very conflicting requirements. Some biosimilar medicines would go through the full procedure as originators, and others would go on limited procedure, limited pathway. And there are examples of that, such as, well, I can share it with you, I can be very sure of that. These are medicines with eminent epoetin alpha and interferon. So the same approach, let's say, our approach with regards to that is that specifically for biosimilars, we cannot see them as generics. So these are not reproduced medicines. This is our official position. And the requirements that we established in 2010 for all products that we review, and so we require preclinical and clinical studies. This is mandatory. Now, what about the situation in Russia with all these registered medicines as of today? The ones that we call biosimilars. Let's talk about that. So these are 9 groups of medicines, basically. You see it on the slide. It's quite a large scope of medicines, but there's the Russian specifics here, different from other countries. Apart from medicines, the state register has a similar procedure for registration of pharmaceutical substances. So not a medical product, but a substance, pharmaceutical substance. So let me talk about it in details. So you see there are 2 diagrams showing the manufacturing process of biosimilar medicines. Now, the first one is a full production cycle. And of course, it goes through all estimated activities with regards to review, analysis, trials, etc. But the second approach, it's not a medical product but a substance. So, and it goes into the registrar as a substance. So, further future steps related to changes in the production cycle, production facility, or changes in the mix, etc., done only with regards to the quality of the product and nothing else. So we believe that there should be mandatory preclinical and clinical studies, comparative studies done in both situations. Now, the applicant for a biosimilar product registration should, on a step-by-step basis, prove a biosimilarity of a medicine with regards to the original medicine, of course. Now, the first thing to do is to prove the quality, similar to any other medicine. Second stage is to confirm a comparative quality. Again, benchmarked against the originator, such as validity, immunogenicity, impurities, purity, mix stability, and stability throughout the lifetime, up to expiry date. Next one is a step-by-step clinical safety and efficacy confirmation, including comparative studies. And the third stage is, of course, a step-by-step clinical safety and efficacy confirmation, including comparative studies. So what are the key issues today in the Russian Federation? The first one is legislation. We're very much hopeful that this will be resolved as soon as possible. Now, the second issue is quality. I'm speaking about the approaches to these groups of medicines developing unified requirements. The third issue is the volume and the scope of preclinical and clinical trials before the decision is made. And, of course, the development of scientific guidelines. And let me very briefly, in a nutshell, show you the algorithm of the way we do a peer review for analogs, for biosimilar products that I would like to exist in the Russian Federation. This is the way we now approach all biosimilar approaches. We're screening for this algorithm. And of course, the stage 1 is comparative quality studies, including statistical biosimilar and the original medicine. It is based on origin, purity, biological activity, and physical chemical properties. The second stage of this review is to determine the scope of preclinical trials depending on the results of the stage 1, and also depending on the properties of the molecule itself as well as pharmaceutical group. I believe that the minimum scope should include the following. So there should be in-house PK and PD studies done in vitro and in vivo, also in-house comparative pharmacokinetic studies, if applicable, and also in-house comparative toxicology studies, subchronic toxicity, assessment of local irritative effect, and impact on immunology. And the third stage is defining the scope of clinical trials depending on the results shown during stage 2. So as you see that in our scope, we're very much aligned with the European requirements. So they're very, very similar, and we've seen it in previous speakers. So I would like to show you the whole process, the way we compile the guidelines. The thing is that the whole concept and the idea of a new guideline is currently based on day-to-day practice. This is current peer-review practice, this is regulatory practice, and we also borrow a lot from international best practices. Now then, of course, this draft becomes part of the agenda for the working group, the working party. And this party consists of experts, scientists, as well as laboratory workers, technicians. So as of today, speaking about our center, we have actually 3 of these working parties, including the working party on biosimilar products, also on reference standards, and another one on cell cultures. And there's also another set of documents. These are documents that have the regulatory act status, which is pretty much a federal law. This is the government pharmacopy and the official outline that we give to the Ministry of Healthcare. So these documents are subject to public hearings, public review, and only then the decision is made on giving them the status of a federal law. Now, as of today, that is, as of 2013, the Scientific and Research Center that I represent have developed 2 overarching guidelines for biosimilars. So these are published in public domain. You can buy it. Of course, we suggest you do it. And these 2 guidelines regard the quality of both preclinical and clinical studies of these biosimilar medicines. And also, which is the most complicated issue, they cover the scope of immunogenicity, assessment of immunogenicity. I think this is the first guideline, and we really worked hard, and the authors of these guidelines really worked hard. There were a lot of consulting on each and every issue, so I believe that the market participants can trust these guidelines and can use it in their everyday practice. So what are the prospects here? What can we expect in terms of publishing in the future? So we would like to prepare 9 specific guidelines for different 9 groups of biosimilars that you see here in the slide. Now we are also drafting 6 guidelines for biological products, and then we will also draft another 9 guidelines related to drug efficacy and preclinical trials with respect to 9 groups of biosimilars. I will also elaborate on another 6 additional complementary guidelines that will be auxiliary. So what are the challenges for biosimilar products as of today? What are they? What do we face today? What are the issues? Well, the first issue, of course, is their legal status. specifically IP-related issues. I mean, be it a developer, be it an expert, and I'm speaking about the exclusivity of data. This is the thing to decide on as soon as possible, because these are complicated molecular structures, and the manufacturing process is also high-tech and very complicated. A third challenge is the fact that there are differences in regulatory requirements and the so-called regulatory inertia that exists. And the fourth challenge, I think that I can put it as follows. I think it's time from development to market. We do understand that yes, these groups of products should get on the market as quick as possible, but at the same time they should do it safely. Don't damage the patient. This is the overarching rule. And finally, to wrap up my presentation, let me also say something about our plans. There are two types of plans. There are short-term and also long-term measures. So let me talk about it. Now, the short-term measures would include the following. This is publishing of guidelines and references and recommendations. So as you see, we're doing this and you will see some results, and we will intensify it in the future. And in terms of the long-term measures, it's about developing a sustainable regulatory system, such as there should be some sort of association that will be dedicated to biosimilar products, because it's a general issue. Russia, United States, Europe, it's all over the place. It's a complicated matter, but from the viewpoint of a manufacturer of these products, requirements are supposed to be unified in all countries, because drugs can have different quality attributes, but at the same time they should be always safe and effective. Thank you very much for that. So, thank you, Alexander, for a very detailed presentation. And now, let's begin our 20-minute Q&A session. So, if I may, my first question is as follows. Tell me, please, are bioanalogies freely interchangeable with the original product? And if yes, in what way? From what I know, in the European Union, the products are not interchangeable. You know that EMA is not regulating the issues of interchangeability. So if you look at the guidelines, nothing says there about interchangeability. But still, as I said in my presentation, there is the so-called automatic interchangeability or substitution, which can be mandatory through local decrees or instructions, through local healthcare regulators or national regulators or governments. And there is also a prescribed one, the one that is backed by clinical trials. So this interchangeability, we haven't seen it, the clinically supported interchangeability, but as I've said, there are products that are currently in phase 3 of clinical trials where interchangeability is tested, and these products will be on the market very soon. So now, a question that we expected. Should producers of biosimilar products that were registered as generics then re-register it after a new regulation becomes effective on biosimilars? Now, the guidelines are all recommendations, and they're not mandatory. They're the procedure on the withdrawal or a re-registration or a pharmacovigilance plan, surveillance. This will be done jointly with the Ministry of Healthcare, and the decisions will be made at the level of the Ministry of Healthcare. The procedure is laid out, and according to the procedure, the product can be withdrawn from the market due to safety or reliability issues as part of the pharmacovigilance plan, and the decision is always with the ministry. So, and we participate in this work. So I believe generally that there will be more details on this, and I think that you will be getting information on that quite soon. Now the next question, what training or retraining should be done by the FABU, which is the official center? Well, the employees of our center get training here in Russia and abroad, and they visit our workshops, laboratory sessions, and the lab workers that participate in laboratory trials also get training abroad, and quite soon, as a matter of fact, we're going to have a visit to the Power Lerlich Institute, where we'll be discussing these issues. We also discuss it with USP and ASAPs. So we are quite proficient, so don't you have any doubts about the qualification of our scientific and research center. Now, the next question I'm getting is, are they prepared to investigate the new classes of medications? I think that they're mostly well prepared. It's my personal viewpoint, nobody else's. And the manufacturer is the one to ask, because the manufacturer is behind production of these products. And of course, these methods that we use in the production process should be delivered to our knowledge, and we should be collaborating on that. But with regards to certain products, there could be issues, and I'm not concealing that. And as part of the laboratory trials and expertise, we'll have to address other laboratories. We're collaborating with other laboratories, so we'll work out all the know-hows available in this area. Next question is, what guidelines or recommendations will you give us for dossier submission process? In terms of dossier compilation and submission, which is a painful issue, the guidelines were delivered, as a matter of fact, 2 years ago. They're still in the process of refinement. Hopefully, we'll very soon submit it for approval to the Healthcare Ministry. The thing is that this issue has been raised multiple times, and as of today, from what I know, the expert conclusion framework has changed. A number of professional market participants and stakeholders do not agree with it, but hopefully it's at the evolutionary track, and we're heading in the right direction. I think that as of today, in terms of the gap and the understanding of the process, and in terms of granularity of the whole laboratory expertise, I believe that we are right on track. We don't have any gaps from other countries. I think that we are basically in a similar situation compared to any other countries that sometimes have day-to-day issues with certain groups of drugs. My question is to Alexander Kudrin. You said that in certain cases of registration of biosimilar products would be enough in terms of comparative PD and PK studies. So phase 3 is not always required. So when do you decide whether there should be phase 3 or not? Maybe my presentation wasn't clear enough, but in all cases we do require phase 3. Always. What I meant in my presentation is that in the future it could be that there will be recommendations at the level of pharmacodynamics studies. So I give you examples where this could be possible. And I was speaking with regards to monoclonal antibodies for malignancies, because they have a very complicated design for stage 3 to understand the survival profile and the progression-free survival, which is very difficult to design. And these could actually exceed a sample size of more than 1,000, sometimes more than 1,500 patients. So in the future, we do expect that there will be PK markers and studies based on these cases. If a company submits enough clinical scientific evidence to support the principles for this study, in that case we will of course consider their options. But so far, everything that we've seen so far, everything that's been submitted to us as dossiers, everything calls for stage 3 of clinical trials. In that case, I would like to add to the above My name is Anton Barzanits from Generian Company. For certain guidelines of EMA with regards to blood, some say that you can be sufficient with PK and PD without therapeutical bioequivalence of stage 3. So how can you comment on that? And specifically in a country where studies between biosimilar and originator studies will be, so what if this originator product is not available? Can you substitute this with a different reference product? Or will be design study in that case? Okay, specifically on the first question, blood products, which is for hemophilia, and these classes of products, right? These products are not the basis for biosimilars and are not covered by EMA legislation or EU legislation related to biosimilars. So these is a different class of products, and these are guided. These have a totally different regulatory pathway. No, I was talking about recombinant blood factors. For recombinant blood coagulation factors, there is bioequivalence principles, but we haven't seen it yet because their patents are still valid for another 10 years. So we haven't seen any biosimilars for these products because they're brand new and they will be on the market for another 10 years. We don't see any expiration. We're not addressed by any companies. They don't submit any biosimilars. So why would you do a biosimilar where the patent is still valid for another 10 or 15 years? It makes no sense. That's number one. And number two, should there be a submission? Should there be an application? In that case, it could be a particular disease that this particular product targets. And for that, we will request a PK and PPD study, most likely, without any pharmaceutical bioequivalence, because it's got to be a smaller size trial compared to the scope that we're currently doing for monoclonal antibodies, because these are products that are registered for the so-called orphan groups. These are very rare, these diseases, and in Europe we have a dedicated legislation that covers these orphan groups, these very rare cases. So everything is on a case-by-case basis and on level of a scientific advice recommendations. So that's all I can say at this moment, at least. I don't want to go into details of the design here in terms of the way the study will be done. It depends on a case-by-case basis. Now the second question that you asked with regards to the product that this is a national regulation issue, and also about the orphan diseases. That is, you issue biosimilar, but the original product, the reference product, let's say, is registered but is not available. So it's registered in the country but is not physically present. What would you recommend in that case? Because we need to benchmark it against a product. So, but if the product is not available, what do you do? Okay, in that case, unfortunately, I have to say it. The only way is a clinical trial, provided that the Ministry of Healthcare would allow this reference product for use in Russia. If not, then some sort of data must be used from patients, patients on which this product was tested, and or any other product which would make this comparative study simple and straightforward. We can clearly see that this research was done in Poland, and the patients in Poland can be compared to patients in Russia. So it's purely pragmatic sort of approach where you can take and so it's retrospective. Pretty much, yes. I can't comment officially, but and I can't comment from the viewpoint of Russian regulated, but that's at least only from the European standpoint.
Thomas Schweidenroche, actually, I have a comment and a question, and just concerning the comment, I quickly would like to go back to the discussion around PD markers. At least listening to Christian Schneider, he made it always very clear that PD marker are acceptable in case they are validated. So in other words, that means they are predictive to clinical outcome. I think this is a very important point. On the basis of this, you might be able to show pivotal similarity, to say it like this, okay? The thing now is, for example, if you look in oncology, these markers do not exist. If you look at immunology, these markers actually also do not exist. I think the example that was presented concerning GCSF is quite a unique case, to say it like this, where clearly the white blood cell count really is predictive for a clinical outcome. I think we have to have this in mind. So really determining biosimilarity on the basis of PD is rather the exceptional case than the rule. And in addition to this, I think the guidelines also always request that a safety study has to be done independent on the PD assessment. So, but my question actually would be for Alexander. You mentioned in your presentation that lots of guidelines are coming up now. And I'm wondering, can you say something about the timelines when they will be available? And also, I'm not that familiar, but will there be a kind of a public consultation process before they get effective, or how is this managed with you now?
Thank you very much for the question. So let me remind you that we've issued 2 guidelines. They're in public domain. And let me say that these are specifically dedicated to general issues with regards to biosimilar products and their quality assessment on the preclinical and clinical trials. This is the first one. And the second one is dedicated to immunogenicity of the product. So these 2 guidelines are already available. They're in public domain. You can buy it. You can try it and you can comment on that. So these guidelines have are recommendations at the moment. So they're not mandatory, and hopefully, should there be any comments from the user communities, would accommodate the views, and we will definitely give the feedback, and if necessary, introduce certain amendments and changes to the guidelines. Now, in terms of the potential, and we believe that over the next 2 years, we'll issue at least 9 different guidelines on other types of classes, and at least 6 guidelines for biosimilar products specifically. More questions, please. We still have enough time. Yes, my question will be on low molecular heparin, because judging by the definition, it turns out that low molecular heparin is not part of the scope. But I know that the situation here is very close to European ones, where they're not really considered biological products, but EMA has a guidelines for low molecular heparins. So I was wondering, how would Russia deal with this issue? Now, with regards to low molecular heparins, there will be a dedicated guideline, and this guideline is one of the 9 guidelines that I've mentioned. And yes, indeed, this group of products is not exactly part of the scope, but there will be a proper pathway for it, and there will be further requirements and guidelines on that front. Oleg Granström from Gerapharm. My question would be with regards to our evolutionary track in Russia. So Madam Maximkina during the first session was very right in saying that there's certain regulatory inertia in Russia, specifically with regards to amendments to the federal law. And because of that, we have more time to observe, and we can take some of the best practices out there, because we're not in a rush, because things take slow in Russia. And I would like to ask a question. Are you planning to, let's say, give a regulatory definition of interchangeability of biosimilar products? For example, let's take United States legislation. Quite recently, on a high level, on the level of specific definitions, interchangeability was specifically written down in the Competition Act, where there's a very clear definition of biosimilar interchangeability. I believe that I should be answering that, but I think that it's not really my domain. I'd love to answer it, but I think that we should address it to the Ministry of Healthcare, because this is a pure regulatory issue, and as you understand, I'm only part of it. I, of course, do my analytical part, scientific, pre-expert work, some peer work, but, yes, we contribute to the legislative activity, but we're not a regulator. We only contribute to it. As far as biosimilar interchangeability, well, it's not my scope, not my call, definitely. If, of course, the Ministry of Healthcare would give me that goal, would tell me, instruct me to work out this issue, then, of course, I will be most ready to do that. But so far, it's just beyond the scope of my activity. And we do understand that this is a very disputable issue, as Alexei said, that, for instance, in the UK they have certain legislative prerequisites, but in Europe there isn't. So there you go. Of course, this issue will be considered, scrutinized, and we're not here on the sidelines. We're working as part of the common system, the system of regulation of drugs in the Russian Federation.
So today I'll be talking about the situation in the United States and what the FDA is doing to implement the Biologics Price Competition and Innovation Act of 2009. I'm from PhRMA, which is the Pharmaceutical Research and Manufacturers of America. We're the trade association that represents the research-based industry in the United States, and we have 32 member companies that make innovative products and also are starting to show an interest in the development of biosimilars. So what I'm going to cover today is biosimilars in general, the Biologics Price Competition and Innovation Act of 2009, or BPCIA, the current status of FDA's development of draft guidances relating to implementation of the BPCIA, and PhRMA's comments to the agency on the draft guidances. So as others have mentioned today, the U.S. law recognizes that biosimilars are not generic drugs, and as such, we have established a separate legal framework for the approval of biosimilar drugs that is completely distinct from the legal pathway and regulatory pathway for generic medicines. Even though they are two completely distinct pathways, there are some similarities, and by law, both generics and biosimilars must have the same dosage form, strength, and route of administration as the respective parent products for the conditions of use for which they are approved. The legal basis for approval of biosimilars in the United States is the Biologics Price Competition and Innovation Act of 2009. This law was passed in 2010 as part of the Affordable Care Act, or Obamacare, and thus it has been in place for about 3 years, almost exactly. This law created an abbreviated licensure pathway for the approval of biological products shown to be biosimilar to or interchangeable with an FDA-licensed reference product. According to the U.S. law, the sponsor must show that the proposed biosimilar product is highly similar to a single FDA-licensed biological reference product, that it utilizes the same mechanism of action to the extent that it is known for the conditions of use or for the approved indications, and has the same route of administration, dosage form, and strength as the reference product. And again, this is part of our legal requirement for approval of biosimilars. The biosimilar pathway in the U.S. set up two types of biosimilar product approval. The first is a biosimilar product, and the second is an interchangeable biological product. These are two different types of approvals under the BPCIA. So both a biosimilar and an interchangeable biological product must show that they are highly similar to a single reference product. And then if one seeks to be approved as an interchangeable biological product, they must not only show that they're highly similar, but demonstrate that they are expected to produce the same clinical result as the reference product in any given patient. And that's language that is contained within the U.S. law. Furthermore, the applicant must demonstrate that there is no increased risk associated with switching or alternating between the interchangeable biological product and the reference product as compared to staying on the reference product alone. And again, this is directly from BPCIA. So this provides the legal framework for how an interchangeable biological product may be approved by the FDA. At the moment, this is just a legal concept. We do not have any biosimilar products approved in the U.S., nor are there any interchangeable biological products approved in the United States. Currently, the FDA is seeking to implement the pathway through the development of guidance, and they've done that. They have held meetings with biosimilar sponsors who wish to develop biosimilars for approval by the FDA, but they have not yet received an application for a biosimilar product. The review timelines for biosimilars in the U.S. are 10 months, so we would expect even if the FDA were to receive an application tomorrow, it would be 2014 before we would see the approval of a biosimilar in the United States. The FDA has also signaled that they view the determination of interchangeability as a second step in the process. Therefore, someone would submit a biosimilar application, be approved as a biosimilar, have some time on market, submit supplemental information in the form of clinical trials, probably switching studies in the clinic to demonstrate the any given patient provision, and then the FDA would review that supplement and potentially approve the product as interchangeable. Therefore, in best estimation, we are probably 2 years away from the first interchangeable biological product. That said, the FDA has not yet issued draft guidance on interchangeable biological products, so there is still some uncertainty in the United States about how this would be done from a scientific and regulatory perspective, even though the legal framework exists in the U.S. So these are the 3 draft guidance documents that FDA has issued. These were issued in February of 2012, and they cover quality considerations for development of a biosimilar, scientific considerations for development, and also questions and answers to some common questions about implementation of BPCIA. Last month, FDA issued a 4th draft guidance document that covers biosimilar product development meetings. These are the meetings that a sponsor would have with the FDA as they seek to develop a biosimilar product. So I'll just go briefly through what is covered in each of these draft guidance documents. Again, they're still in draft form, and they're subject to revision before they are finalized. These draft guidances went through a public comment process in 2012. The FDA received a number of comments, and they are legally obligated to review those comments and address them prior to finalizing these guidance documents. So the quality considerations draft guidance covers FDA's view on the comparative analytical studies that make up one component of the demonstration of biosimilarity. So they really view the quality assessment as the foundation of the biosimilarity demonstration and the scientific basis for any abbreviation of subsequent preclinical and clinical trials. The FDA expects that there will be analytical similarities, the same amino acid sequence, molecular weight, complexity, heterogeneity, functional properties, etc. that the applicant will conduct extensive, robust, comparative physiochemical and functional studies, biological assays, binding assays, enzyme kinetics. And that the applicant would also demonstrate comparability in terms of stability through stress studies with both the reference product and the biosimilar product. The guidance document also covers additional CMC information that may be relevant to the assessment of biosimilarity, and then indicates that on top of the demonstration of analytical similarity, there is a need for additional preclinical and clinical studies. And so this brings us to the second guidance document, which is the Scientific Considerations Draft Guidance, and this builds upon the quality assessment that the applicant has completed. And this really starts to set up what FDA calls a stepwise approach, or a totality of the evidence approach, where they look at the similarity that has been demonstrated on the analytical level and then appropriately tailor the preclinical and clinical studies, or abbreviate the preclinical and clinical studies on the basis of the analytical program. So again, just as others have said, the FDA is taking a stepwise approach. The draft guidance includes considerations of the complexity of the product, what type of molecule it is and how complex it is, and then the general scientific principles for conducting structural and functional studies, and also the animal studies and the clinical studies that would be necessary to demonstrate biosimilarity for the FDA's purposes. The draft guidance briefly mentions extrapolation of clinical data across indications, and also post-marketing safety monitoring and risk management plans. The Q&A document is really just that. It's a series of questions and answers, common questions about implementation of BPCIA. The FDA explicitly says that the initial draft guidance documents are not intended to address interchangeability. They apply to the development of biosimilar products, but not to the development of interchangeable biological products. And again, this is because while it's a legal concept in the US, it's not something for which there is scientific and regulatory consensus about how it will be done. So in the future, FDA may issue guidance on interchangeability, but at this point, there is no guidance from FDA on how to achieve or develop an interchangeable biological product. There are also some questions that are really more procedural about requirements to submit a BLA and also reference product exclusivity. So this again just shows the stepwise approach, building upon the analytical studies, additional animal studies, and then finally clinical studies to get that comprehensive program that really demonstrates biosimilarity. FDA does have the legal authority to waive any of these requirements if they feel like it is scientifically justified, although the law does set out the expectation that analytical, preclinical, and clinical work will be done in a biosimilarity demonstration. So in general, pharma is fully supportive of the approach that the FDA has laid out for the development of biosimilar products. We believe that the guidance documents are appropriately high level and overarching to permit biosimilar developers to understand the FDA's expectations for developing a biosimilar product, but also allow appropriate regulatory flexibility as they evaluate these products on a case-by-case basis. And we recognize that that is something that regulatory authorities will need to do, given the diversity of biological products and also the different approaches that an applicant may take to demonstrate biosimilarity. We did urge the FDA to be a bit more detailed in their recommendations regarding molecular similarity and to advise against seemingly small changes to the design of a product which may have unintended clinical consequences, especially since these consequences may be difficult to predict, especially for biosimilar manufacturers who will not have extensive manufacturing experience with the product in the way that the innovator company will have experience with their product over years or even decades. So again, it's important that as you go into the design of a biosimilar product, that you minimize controllable differences. And these are things like amino acid sequence that can be tightly controlled with the cell type that is used for the production of the biosimilar. The FDA recognizes that as these controllable elements are changed, the feasibility or utility of an abbreviated biosimilar pathway may be decreased. So it may be more difficult to demonstrate biosimilarity if there are changes in the design that are otherwise controllable. As others have said, the FDA has recommended using multiple analytical procedures that are sufficiently sensitive to detect differences in the product. So using orthogonal methods to test the same parameter in different ways to ensure that the products are as similar as possible at the molecular level, using state-of-the-art technology, but also recognizing the limits of technology, and to do follow-up work as necessary in preclinical and clinical studies. Regarding preclinical testing, we agree with the FDA that in general the preclinical program should follow recommendations in ICH S6R1, and that animal studies should be done in a relevant species and done according to the 3R principles. However, we do believe that some animal studies are necessary before the biosimilar applicant goes into human studies. So it would be inappropriate to go into human trials in the absence of any studies in animals. So the FDA recognizes that clinical testing is an important component of the biosimilar development pathway, and it is important, according to U.S. law, that the applicant demonstrate the absence of clinically meaningful differences between the product and the reference product. So the U.S. law requires exactly that, that you demonstrate the absence of differences, not the presence of similarity. So it's a bit of a different way of looking at things than perhaps other regulatory authorities. So you need to do sufficient tests to show that there are no differences, not just demonstrate similarity on a set of parameters. And this is really to ensure that patient well-being is protected through clinical development of biosimilar products. When it comes to extrapolation, the FDA has indicated that extrapolation may be permitted, but there needs to be strong scientific justification for extrapolation across indications. Pharma believes that extrapolation across indications is different from extrapolation within indications, and that caution should be used when looking at extrapolation across wholly different indications that use different mechanisms of action. But within indications, it may be possible to extrapolate clinical data if the appropriate scientific justification is given. Regarding immunogenicity, the FDA recognizes that immunogenicity testing is a key component of pre-market testing prior to approval of a biosimilar medicine, because biologics have the potential to evoke an immune response. We believe that the FDA should require that pre- and post-market testing be performed, including preclinical and clinical and post-marketing pharmacovigilance. This is really just a patient safety provision, making sure that adequate work has been done pre-market and post-market to ensure the safety and efficacy of biosimilar products. Regarding labeling, there's a small piece of advice from FDA regarding the labeling of a biosimilar product. The guidance advises that the label include a clear statement that indicates whether the product is biosimilar or an interchangeable biological product. So in the US, the labels of biosimilar products will clearly say this product is a biosimilar, it is not an interchangeable biological product. Or the converse, if it has been approved as an interchangeable product, it will say the product has been approved as an interchangeable biological product. So that is essential information that will be included in the product label. And FDA has recommended this because they believe it's essential that the regulatory approval standards for the product be clear in the product labeling so the physicians understand how the product was approved, and what was done to justify the approval. Pharma believes that this is a good first step for clear labeling and informing prescribers, but it would also be helpful to include information about what exactly was done in the biosimilar program. So what studies justify the indications for which the biosimilar product has been approved. And then finally, pharmacovigilance. This is covered to some extent in the FDA draft guidance. The FDA has recommended that the pharmacovigilance plan be in place at the time of approval, and that in most cases it will look very much like the plan for the reference product. So if the reference biological product has a REMS, or risk evaluation mitigation strategy, the biosimilar product will also have a REMS, probably with similar language, similar scope as the reference product. However, the FDA has not dealt with the issue of drift in draft guidance, and theoretically that would be something that they will need to consider before they seek to set the standards and recommendations for development of an interchangeable biological product. The FDA has not yet issued a position on the naming of biosimilar products, but pharma believes that unique non-proprietary names are an essential feature of a robust system for adequate tracking of adverse events associated with biosimilar use. This again is a subject of debate in the US, although recent approvals of biological products have included a 3-letter prefix prior to a common stem for the approval of two recent innovative biologics. The FDA has said that this is not an indication of how they will handle the naming of biosimilar products, but it is a change in the practice of the FDA with regard to naming of biological products. We also believe that it's important that products have a unique name at the time of market entry. Because of the system in the US where you would first be approved as a biosimilar product, and then after a period of time and with subsequent data may be approved as an interchangeable biological product, it's important that the products are distinguishable at the time of market entry, and ideally do not change names because of a change in regulatory status when they're approved as an interchangeable product. So again, something that the FDA needs to consider as they think about the life cycle of these products and how they may evolve over time. So to summarize, a biosimilar in the United States will be highly similar to, but not the same as, an FDA-licensed reference product. The FDA has proposed a rigorous science-based process for evaluating proposed biosimilars. Pharma believes that unique non-proprietary names for biosimilar and interchangeable biological products is an important component of robust pharmacovigilance, and that the decision to take a particular biological medicine should be made with the oversight of the guidance of a physician, and all of this can be facilitated by clear labeling and unique names for the products. And finally, the FDA and pharma share a concern for the well-being of patients, and also an interest in incentives for continued innovation of new medicines and new access to therapies for patients. Thank you.
Good afternoon, ladies and gentlemen. Thanks to the organizers for inviting me here. I'm really enjoying my time in Moscow. It's actually my first time in Moscow and in Russia. I'm based in Austria and have spent the last 10 years in regulatory affairs for biosimilars. So since the very early days when these new product type came up. So my topic is the challenges and also the experiences that I would like to share with you that we gained over the years. I have 4 topics, a brief introduction, although we heard a lot today already. Then how are these products developed? Then our own Sandoz experience. And I will conclude with some comments on the future of biosimilars. Just a brief introduction. Mostly we have touched on this today, how biologics first came about from the 1950s until the major brands we have nowadays, and the latest research in stem cell and gene therapy. Here's just an overview of the top 10 sellers worldwide at the moment. And as you can see here, already now 7 of the top 10 products are biologics, and also the enormous revenue that these products bring in every year. And this is a trend that will continue, I think, and will also continue to be a huge burden on health budgets around the world to finance these very expensive products. A definition, what is a biosimilar? I don't think I need to go over that. We already heard today how a biosimilar is defined by a stringent regulatory pathway and what needs to be done to demonstrate comparable quality, efficacy, and safety. Also the molecule size we have already seen. Just to repeat a little bit here is to make you see the enormous difference in molecule size. If you look at a small aspirin on the left and move your way up through a calcitonin, filgrastim, somatropin, EPO, and finally a monoclonal antibody with a molecular weight of 150,000 daltons, you can imagine the complex structure and also the challenge for process and analytical science to first produce and then also to analyze and characterize these type of molecules, which really requires state-of-the-art analytical technology. We are working with living organisms, as we already heard from previous speakers, which produce a lot of variants and byproducts, which then have to be purified by a whole sequence of downstream processing, DSP, to finally generate a clean drug substance, which may still be a mixture. For example, the glycosylation of an EPO or a monoclonal antibody is never really just one molecule. It's always a mixture within specified limits that are acceptable. And finally, we need to formulate into a stable and patient-convenient product. Could be in a prefilled syringe or in a cartridge or in a vial. Access to biologics, we also heard today, is a growing issue around the world. And if you look at these examples here, maybe you wouldn't think that in Europe, US, Canada, or Japan, there's any problem with health budgets to finance these products. But there is. For example, in the EU, 1/4 of the arthritis patients do not get treatment with monoclonal antibodies. In the US, there's an example that cancer patients are twice as likely to go bankrupt after the first diagnosis. Or Canadian children with juvenile idiopathic arthritis may not receive the standard care because the coverage for biologics is incomplete or inconsistent. And also in Japan, which you might believe there's a good budget, also there only half of the severe rheumatoid arthritis patients receive the appropriate treatment. Now, what's happening on the regulatory side? We already heard from Ivana about the WHO guideline. Here I just give an overview of other regions in the world that are working or have already published guidance on biological products. So I guess Russia should also be on this map, at least when the new promised guidances will be implemented. You see here a lot of activity in the Latin American region and also in the Southeast Asian region, apart from the ICH countries, Europe, US, Canada, and Japan. So how do we develop these type of products? To give you an overview of how long does it take and what does it cost? The cost can be up to $250 million US dollars, and it can take up to 8 years to develop a biosimilar, because the first 2, 3, or even 4 years would be spent just on molecule characterization and process development, since the biosimilar has to develop its own unique process. The technical challenge is to really get to a highly similar molecule and also to develop a process with an acceptable yield. Finally, in the clinical trial part, it's the task to confirm highly similar efficacy and safety, to support extrapolation across indications with all the difficulties that we've already heard today that need a really sound justification whether or not you can extrapolate from one indication to another. It may not always be possible. And also the speed of patient recruitment and trial execution is something that can slow down a biosimilar development a lot if you can't find patients quick enough. As an overview here, how is the biosimilar development structured? First, on the bottom left, you start with the target definition, like which reference product do we want to create our own version of. Then number 2 on the top left is the target-directed development. Another speaker earlier today said it's no use to just think you can transform an existing development into a biosimilar development. You really have to be sure from the start what you want to do, that you intend to develop a biosimilar, which then starts with the recombinant cell line development, bioprocess development, and finally the purification process development. The pyramid is upside down here because the target range becomes more and more narrow, more and more specified until we reach the drug product development. And then we finally design the target range for the biosimilar. On the right-hand side, the pyramid, the other way around, which symbolizes that you have to do this development step by step, starting at the bottom with the physicochemical and biological comparability. Not like some companies may intend to shortcut the physicochemical part and just do a phase 3 trial without fully characterizing their proposed molecule. So you really have to work through each step and only go, for example, into preclinical development if you are totally convinced that you have a highly similar molecule in hands. And again, then, if you did find differences in the preclinical development, you would have to go back to the lab and change your molecule before you go further into the PK/PD human development. And then finally, phase 3 at the top would be the confirmation of efficacy and safety. I should say a confirmation of comparable efficacy and safety. As we also heard already earlier today, the biosimilar does not have to prove again that the product is efficacious, or efficacy per se is not needed, but rather a comparable efficacy to the reference. In this pyramid on the right, the clinical part seems very small, but in terms of cost, of course, the phase 3 would be the most expensive chunk of the biosimilar development. So how long does each step of the development take? For preclinical, we calculate about 6 to 12 months for the abbreviated biosimilar program that Hans-Karl Heim from BfArM described today. In the phase 1 part, it takes about 9 to 12 months to demonstrate PK and or PD equivalence in a sensitive population. It can be healthy volunteers, but for some products or some indications, it's not considered ethical to go into healthy volunteers, so phase 1 would already have to be in patients. Phase 2, the dose-finding studies are usually not required for a biosimilar because biosimilar uses the same dosing regimen as the originator. Then the phase 3, a confirmatory trial, can take 2 or even up to 4 years because of first the patient numbers would be several hundreds. A common figure would be about 500 patients in a biosimilar trial or more. So any company submitting a dossier with just maybe 20 or 30 patients in phase 3, which does happen, I believe, would, I think, could not be considered a serious application for a product. Finally, the post-approval trials is usually a requirement that authorities commit the applicant to do even after approval. Sometimes post-approval safety studies are undertaken, or non-interventional studies in indications that were initially not studied during the registration studies. So the development work continues even after approval. Now to our experience that I'd like to share. The analytical methods to study the structure leads to, or really needs high resolution analytical techniques. Orthogonal methods were already mentioned today to look at one molecular peculiarity with different methods. Only if you really apply the full battery of state-of-the-art, most modern protein characterization methods, you can reach a full understanding, or at least from current point of view, a full understanding of your molecule. I will not go through the details here, but you can see all the molecule attributes on the left that we need to study. And on the right, just an example of different methods that are commonly applied to protein and also monoclonal antibody characterization. In the middle, I'm just summarizing that the primary structure, of course, needs to be the same and needs to be verified that it is the same as the reference. The glycosylation profile can also be comprehensively studied. And on average, there's even up to 100 different results, or 100 different methods that lead really to a complete picture of the molecule characterization. Now I'd like to go to a topic that was also led to some debate earlier today about manufacturing changes that happen. This is a slide borrowed from Christian Schneider, who was also mentioned today already. He's has one of the lead positions in EMA. Here you see some statistics on how many manufacturing changes have been approved by EMA for these originator products. Like for Remicade, even 37 manufacturing changes since the first approval. I mean, these changes are not, the full details are not disclosed, of course, but some details are known. For example, that maybe the suppliers of a cell culture media change, so that would have to be submitted, or there are new purification methods which are submitted, or there's even a site transfer to another manufacturing site, which of course is also submitted and approved by the regulators. So what does it mean for the biosimilar? This variability that we can observe in with many originator products, and there was also a question today to our colleague from Roche about this publication here that investigated certain quality attributes of rituximab and other molecules over several years and determined that there were changes in ADCC potency, for example, and that a pre-shift and post-shift quality could be clearly identified, really proving that there was a significant change to the product. Here in this example, it was actually a shift in glycosylation pattern, which then resulted in a different potency, which could then be measured with this ADCC assay, which probably is an indication of a change in the manufacturing process. And we have actually monitored these things happening in several originator products. So this variability really provides then the goalpost, so to say, for the biosimilar development. So where we need to get with our target range is determined by this variability. These goalposts are very narrow. If you look at the green circle, that symbolizes the variability of the reference product. The yellow circle is the variability we observe during our own cell line development before we select the best clone or the most appropriate clone. And then the red large circle is the theoretical or biologically possible variability. So the green circle is really quite small, what we're talking about, but still the analytical methods of today can differentiate between these different product qualities. And this admittedly small variability then sets the goalposts for the biosimilar. As you can see here, in a different view, the goalposts again of the originator material before and after the change, which is then, from a scientific point of view, also the basis for extrapolation of indications. If you think about it, like if this variability was scrutinized or not approved by the regulators, then then also the extrapolation of to other indications of the originator after manufacturing change would be in question. So in the other way around, or in other words, since this variability is happening, has been happening for a long time, and also regulators are used to reviewing and approving manufacturing changes, this in return is the basis for the extrapolation of indications for the biosimilar, and also for interchanging one product for another, because the biosimilar sometimes gets closer to the originator than the originator is to itself. If you look at those goalposts, if the biosimilar is well within these goalposts, then there's no reason to reject interchangeability for a biosimilar. If our variability is even smaller than the variability of the reference product. I know we have heard that different authorities around the world regulate interchangeability differently. Some of them leave it to the member states or to the physicians, but this is really the scientific explanation, the scientific backbone, and also the whole purpose of a biosimilar development is to interchange the biosimilar with the originator product, which is documented and well proven over all those 7 to 8 years of biosimilar development, including a huge amount of data. So the quality attributes of a biologic that we have to be careful with can be influenced at all stages of cell line and process development. Just some examples for you here, what can happen or where the sensitive areas are. For example, the host cell line or the transfection and amplification pool, the genetic setup of the production cell line and the clone, or the growth medium composition is important, the culture conditions, the upstream processing type, the culture history, the individual drug downstream processing steps, the hold times of any intermediates that you might have in the process, how long can you store them, and then also the storage conditions of your buffers, of your containers, your solutions. So all these factors are very important to consider in your biosimilar process development or any biological process development. You might have heard the term quality by design before, which is also documented in various guidelines. Here the attention to detail is very important. For example, here the characterization of a monoclonal antibody. This is a high-resolution identification and quantification of major and also minor glycan structures down to a level of 0.1%, which is important to also resolve and characterize even the small peaks to know exactly what is in your product. And the second example on the lower half shows you all the theoretical possible ADCC results that you can have with different clones, like all those colored squares in the bottom left graph represent different clones. So this really shows you what is possible in terms of ADCC or in terms of potency of the product. And on the right also, it symbolizes the iterative process of how to get to the correct or the best clone from a polyclonal mix, finally to a monoclonal component on the right, which is the blue set of squares, which is in this case the desired clone. There are products around, I called them here follow-on biologics, that would not deserve to be called biosimilars and would not be approved in EU. This is an example with Reditux, which is made by Dr. Reddy's in India, and compared to Rituxan, compared to the originator product, where you can see at the black line a lot of additional peaks that should not be there, that are not found in the originator product, which are a sign of probably a higher host cell protein content, or of aggregates that should not be there, that are not comparable to the reference, and also different glycosylation profile, and a non-comparable ADCC effector function. If you look at the clinical data that were submitted and later published with this submission, only 17 patients were in the PK/PD study. So this is kind of the opposite example, how it should not look like. I have another example along similar lines in an isoelectric focusing gel. This is an older publication now from a Dutch professor that sourced different EPOs from around the world and compared them just with a simple gel electrophoresis method, where you see on the far left and right, under E is the reference, and all the samples from 1A to 8 are these products from different countries, which are all have additional bands, which are clearly a sign of variants or process-related impurities that should not be there. On the right, those two prints is what it should look like. Here you see a biosimilar EPO and its reference product Eprex, approved in the EU. Here you see no difference in this gel between the two products. The biosimilars must match the originators across multiple quality attributes. It's like a fingerprint, which is also unique, which can be achieved with the whole set of analytical methods that are nowadays used. I'm just going through this quickly to show you what we're looking at. Of course, primary structure, higher order structure, biological activity, impurities, post-translational modifications, using all these different methods to finally get to a full fingerprint of the molecule and fully characterizing the molecule with currently available state-of-the-art methods. And then also these attributes can be combined, and new knowledge is gathered from the combination, from the totality of the data and totality of the evidence that is generated on an analytical level. Yes, as mentioned before, there are many methods applied which in total deliver up to 100 different quality attributes. Some examples of our clinical experience with approved biosimilars. First one is somatropin here, where we already began our clinical studies 12 years ago, long before EMA came up with official guidance on biosimilars. We had a very long-term study that kept going for 84 months, since growth hormone treatment is a chronic treatment. We continue to treat patients and gather safety data, which now resulted in a safety database of 35 million patient days of safe use with our growth hormone. Also for filgrastim or GCSF, we now dispense of 3.5 million patient days of safe use and are in the market since 2009. And finally, also for EPO, epoetin alpha, we have treated more than 5,000 patients and have generated 67 million patient days of safe use with the product. Now, finally, my conclusion is that biosimilars already expand patient access today This is an example from Great Britain, where the use of G-CSF before the advent of biosimilars was actually decreasing. Before 2008, every year there was less G-CSF used due to the cost. Once biosimilars came in, the use was increased again, and many physicians moved G-CSF treatment back to a first-line treatment, or back to a prophylactic treatment. And before that, it was only like a second line or reserve treatment, which makes a lot of sense, I think, because G-CSF also prevents the rehospitalization of patients if they catch an infection after having been treated with chemotherapy. So here biosimilars have really contributed to an improved patient access. Just to summarize on my final slide, biologics can nowadays be thoroughly characterized and understood by modern state-of-the-art technology. And they can be designed and manufactured to fall within the variability of the originator products and are or can be created in a highly similar way. The clinical trials are only there to confirm the comparable efficacy, but do not need to establish de novo efficacy again of the product. Many years of experience now have demonstrated that EMA-approved biosimilars are as safe and as efficacious as the reference product, and EMA has surely the most experience of any regulatory authority in the world with regulating biosimilars. And biosimilars are already now broadening patient access and are improving the affordability of these still quite costly treatments. So, thank you very much.
Thank you. Thank you for the introduction and the invitation to be here to present and share on this both exciting and always interesting topic. I think I actually have a tougher challenge than Thomas Schweitmüller did before lunch, because I'm not what's between you and a reception. So I will go directly into my topic, comparability and process changes. Of course, I also have the luxury that at this time of the day, after all the presentations, a lot of the material you will have seen before, so I can be briefer somewhere. And actually, this slide is very similar to what Thomas just showed in the previous talk. But there are a few highlights I want to make here. Basically, that the background for the manufacturing of biologics, why each process is unique. If we look at chemical entities, if you read the patent, you read the structure, and you get a method of synthesis that basically you can go out in the laboratory and you get the same result. If we look at a patent for a biological product, for a protein, the patent describes a method for developing the DNA and a method for cloning that DNA into a cell system. But that's where it stops. The cell line does not fall out of the patent. So this is again where you have to start from scratch, synthesizing your DNA and make a lot of decisions. What vector you use, what cell line, what plasmid, what host cell you use, what kind of cell culture system, what kind of purification, and so on and so forth, and analytical methods. And whereas some of this information for the originator product is available in the public domain, it is, I can guarantee you, far from all. There is a lot that is proprietary information. So this is where it ends up that you will, of course, if you make the same protein, you will encode the same amino acid sequence, but there can be DNA differences. You're likely to work in a different vector, in a different cell system, in a different, using different in-process controls, because these need to be adapted to the way you run your process. different purification methods, different formulations, and different analytics. So there's actually a lot of source of variation, and that is regardless of whether you are developing a new innovative process from scratch or a biosimilar process where you want to mimic another product. And this, again, it's already been emphasized, but back to what Dr. Kudrin said, that you really have to make up your mind from the beginning. If you want to mimic another product and make a biosimilar, you have to start that train of thinking right from the beginning, where you synthesize your DNA and put it into a cell line. Another slide you've seen most of before in Dr. Pini's presentation, specifications are linked to the entire manufacturing process. I think this is a very important slide because, again, it illustrates that you cannot just look at your final drug product and the specifications for that. It really starts with the cell bank, with the processes, drug substance, and the way you develop and control drug substance and drug product processes. So there are all these specification elements tied into your product, and it shows how the process and the product are tightly linked. Furthermore, for the specifications, also the product that went into the tox studies and the clinical studies are, of course, very important. These are part of the basis for what forms the specification. So, a comparability exercise. It's already been mentioned that this is something we have known. It's a tool that we've worked with for many, many years, came along a lot before the biosimilars. And what I do want to emphasize here is that it is a tool we use to control manufacturing changes and make sure that the product after a change is as safe and efficacious as before. What I do want to highlight is that manufacturing changes, we do lots of them, was also indicated in Thomas's slides, but also that they actually come in a variety of forms and sizes, so to speak. So there's anything from what's very, very small, what's indicated in the green end of the spectrum here, small changes, low risk, they're frequent. It can be change of the supplier of sodium chloride or whatever. It is a change, it's a small change, it can be documented by chemical analytical control methods. Then we can go to the red spectrum, still within the same process, and look at high risk changes. These are rare because there's a lot of work involved, and that, as we all know, takes both time and money, and means that the resources you use for that, you cannot use for another drug development program, for instance. Examples here can be you need to change your cell line for whatever reason, probably productivity. It can be a new process, it can be a new formulation. So these are sort of at the other extreme. And clearly when we are out in that red end of the spectrum, clinical trials are needed to support such a change, also for any product, also for an originator product. What's also important here to realize that when you look at biosimilars, once they are approved, as has been mentioned today also, then they start living their own life, and they will control their manufacturing changes in exactly the same way as an innovator product. If we then move to the right part of the slide, there's a gap. And that's because if you develop a biosimilar, there will always be a gap to the innovator product because you don't have the same cell line, you don't have the same this and this and that. So it's always a high-risk change. Clinical and non-clinical data will always be needed, as also laid down in the guidelines both for US and Europe. So how is a biosimilar designed? Well, basically, you start looking at the reference product and sort of make that target, and I think Thomas has actually covered that very nicely, so I don't need to go into that. But what's also clear here is that you need all those elements, the entire module 3, you need the physical, chemical, biological characterization, and the preclinical and the clinical trials. And I think what comes out of it, if we look at some sort of a design space, the shape I've aligned here is the space, the attribute space, can you say, for the innovator product. And I think what's important here is, of course, that that's not a two-dimensional space. It's rather a multi-dimensional animal. But this is what outlines the total experience based on what we have: clinical experience, manufacturing experience. So this is sort of the space that where we have agreed with the regulators that we know what's going on and it's okay to be here. So some of the changes that we have discussed today, where yes, we move around, we can see a difference before and after, but we're still within that sort of overall known sphere, shape, what have you, so it is still okay. If we then look at a biosimilar, I think we can be very confident that the target is very well defined and a lot of that shape will be the same. But I think there is still the open question, because these are biological systems and unique systems, that there are so many elements here that all of the periphery, all of the shapes may not be completely superimposable, and that's of course what we need to focus on, as also clearly demonstrated in the guidelines, that you have to look for differences to establish, you have to design your system so that they can identify differences in order to establish the confidence that we have achieved similarity. So this is another way of looking at a comparability exercise for an innovator product in blue and a biosimilar product in red. For an innovator product, when you plan a change, you basically analyze what does that change imply, look at the science behind it, what kind of a change is it, and do a quality risk assessment. And then at all the subsequent steps, you do the proper analytical development and monitor and see what happens, and do you then have comparable data before and after the change. It's clear if we compare a biosimilar to an innovator product, we don't have all that upstream data, so to speak. So that's why it is important and necessary to supplement with the non-clinical and clinical comparability exercise, so we in that way cover the whole area. So from there, I would like to just share a few examples of process changes to give some more flavor for what is it actually we talk about. I would like to start with insulin. After all, I am from Novo Nordisk, and insulin is near and dear to our hearts. And I also think it gives some very interesting or relevant illustrations of what we're talking about. On this slide is outlined the process characteristics for two innovative insulin products: Eli Lilly's, that is produced in E. coli, and Novo Nordisk, that is produced in yeast. And simply because they are produced in such different expression systems, that has impact for the way the disulfide bridges are formed. There are differences for the way the products is expressed, is purified, and is thereby also analyzed. So although it is the same sequence for human insulin, the way to get there is very different, and that does have an impact on the potential for the impurity spectrum that is relevant for each of the products. So if we look at this slide, these are the derivatives that originate from fermentation in a yeast-based cell system like ours. And that picture will be different in a coli-based expression system like Eli Lilly has. These are further impurities that can be seen further downstream in our purification process because of the way we move from a single-chain pro-insulin-like structure to the two-chain insulin structure. And that is closely linked to the way we have designed our precursor and end up with the final insulin product. And that brings me to another important point, looking at pharmacopoeia. Pharmacopoeia are great tools. They highlight some of those characteristics that we can and should expect in a given product that is listed in the pharmacopoeia. But of course, it focuses on the known characteristics that all these products share. So complying with pharmacopoeial methods is of course a must. But there is no way that this can replace the whole comparability exercise, because the pharmacopoeia methods are not designed to look at the differences that you can expect between products produced in different ways. So another example from moving to a process change for one of our insulin products. And this is actually a case of one of the more drastic, high-risk changes, if we can call it like that. For capacity reasons, we needed to expand our capacity quite significantly. And to do that, we changed our production cell and put in new DNA that basically allowed the cell to be more high-producing. And we optimized the fermentation process, recovery process, purification process, and built a new facility. So as you can see, a very comprehensive change. And the way this is documented was by a full CMC package and comparability exercise, of course, where we could see minute differences in the impurity profile, but differences that we could explain, and in compounds that we already knew. To support that, of course, we needed to do clinical work. So we did PK, pharmacokinetic, pharmacodynamic studies to show that the insulin efficacy was comparative. And we also did immunogenicity studies because, again, as we've talked about a number of times today, immunogenicity is one of the safety areas of concern. So even for a small product like insulin, clinical safety studies are necessary to demonstrate the absence of clinically meaningful differences. Another important product or important change I would like to share with you is one we investigated for our recombinant factor 7A product, which we have expressed in a BHK cell line. We have explored moving to a CHO cell line because that has some more robust manufacturing characteristics. And again, we did a full comparability exercise. We showed a lot of similarities between the two products, but we also showed that there were differences in the glycosylation. As already discussed today, glycosylation for mammalian cell products is something that is very, very important. And it turned out that when we put these two products into a human PK studies, we actually saw a significant difference in the area under the curve, a difference of about 30%. So a clear example of a change that did not lead to a comparable product, and where, for obvious reasons, we could not pursue that further to just as a normal process change, so to speak. So this change in the cell line really led to a significant difference. We have not at this point in time taken that forward and studied in safety studies, but for a product like factor 7, it's a coagulation factor, it is a large protein. Obviously, immunogenicity is also an important aspect that needs to be investigated if one wants to implement a change like this. Myozyme made by Genzyme is another interesting example. They did a scale-up. Myozyme is also a large protein produced in CHO cells, originally in 160 liters, later scaled up to 2,000 liters. This change or these files have been reviewed by both EMA and FDA. They come to slightly different, actually they don't come to different conclusions, but it's managed in slightly different ways. In Europe, only the 2,000 liter material is approved. I assume that it was submitted later in Europe, so that was the easiest way to come across the differences they saw, because they did see differences in the impurity levels, and they saw differences that raised a concern for immunogenicity differences, and that's why in Europe they only approved the 2,000 liter material. In the US, the 160 liter material was already approved, so when FDA reviewed this process change, they basically decided that this was a new product because of these differences, so they approved it, but as a new product and with a distinct new trade name. again, it illustrates that you really have for proteins of this character, changes do have an impact, and it needs to be managed carefully. A few biosimilar case studies before my time is up. And here I do want to just take a look at this overview of the European guidelines again, basically because I think it's a very, very useful set of guidelines developed in Europe with the quality and the non-clinical and clinically, and especially with the product-specific guidelines that I think are very helpful, again, because they focus on the disease area. As already mentioned, it doesn't really, when we look at the clinical program, it is not important if we're studying a protein or a chemical entity, it's really about the disease. So these outlines the comparability trials that you should design for these various products in those areas. And I also here want to actually to highlight that we talked about before, that sometimes it is enough for efficacy to study PK/PD. There's actually one more example here on this list, and that is for insulin, because for diabetes, HbA1c is a validated surrogate marker to demonstrate efficacy. So for a biosimilar insulin to demonstrate comparable efficacy, PK/PD studies are adequate. We still need immunogenicity trials, but for comparable efficacy, that is enough. So the principles of the biosimilar approach that has been covered, so I'll move on. And also you've seen this list already today, and I think, actually this is the list of the products that have not made it, but I think the list you've already seen today demonstrates that the European system works and works well. Products get approved. Sometimes products don't get approved, and if you study the EPAs, I think it's very clear that there were good reasons for that these products did not make it, whether they were withdrawn or rejected. It's interesting to see on this list that, again, insulin is actually here in two rounds, so to speak. And it again illustrates that although you would think that a small protein like insulin, that it's easy to do, it doesn't seem to be so easy after all. And as a company, we've been working with insulin for 90 years, so I think we understand some of the challenges that people come across. So this is one of the examples of differences I would like to share. This is from the first round of the Marvel submissions, where they again looked at pharmacodynamic data to demonstrate similarity, but actually what they did demonstrate was not similarity in the pharmacodynamic profile. They also did not demonstrate that the purity was comparable to the reference product. So again, it's a stepwise approach, as I think is also very clearly outlined in the US guidelines, and I think that's very sound. If you don't achieve chemical comparability, then you should probably do something else before you start doing the clinical trials, because you are likely to hit serious problems in achieving your goals in that way. So this is a list of approvals. Again, we've seen that, so let's move on. And actually, time is running. Rather than going into more details on these these products that did not make it, there's one other example I would like to share, which is a product, this is from literature. It is a somatropin sourced, I don't exactly remember where, but it's a group that has worked with analyzing a number of somatropins and actually found a new thioether variant in one of the products on the market outside Europe. They also investigated further to find out what was the chemistry behind this variant, what sort of conditions would lead to such a variant. And what they found out was that this variant was generated at high pH and elevated temperatures. So obviously something you need to look out for when you design your process. Proteins do not always like combinations of certain temperatures and certain pHs, it may impact the profile. What is another important learning from this example is that this new compound was not picked up by the pharmacopoeial methods. Again, the pharmacopoeial methods have their advantages, but they are not designed to assess changes of this character. So basically, this group found this new peak probably by mass spec, and then they worked backwards and found an analytical method that could quantify it reliably. And actually they found out that it had a reduced biopotency, so definitely something you will want to think about when you produce growth hormones. So conclusion, products must have highly similar molecular structural features. We've already talked about that. Impurity profiles must be highly similar. So again, the quality comparability really must show comparability, otherwise make that happen before you move on and go into clinical studies. And then I think what is also nicely captured in the US legislation, it talks about you have to be able to exclude clinically meaningful differences in efficacy, safety, and immunogenic potential. And that basically means that, yes, we need to confirm that we don't see anything unexpected in the clinical trials. So with that, I'll conclude, and thank you for your attention.
Thank you very much, Inga. That was very excellent and extensive presentation. And I'd like to open now our Q&A session.
And your questions, please. Yes, and I have the following question. So, it's a question to both Thomas and Inga. So, before submitting a dossier for biosimilar product registration, do you have any consultancies with the regulatory authorities, specifically in order to define the size and the scope of analytical procedures, or is it actually sponsored by the company itself? How do you define the scope of analytical procedures to prove and demonstrate biosimilarity? This is the question for both companies.
Okay, thank you. Yes, we do approach the regulatory authorities, usually at the time before we go into the clinical development. Like we would have already completed the physicochemical characterization, but before really going into the very expensive clinical trial phase, we would want to make sure that regulatory authorities agree with our program and also kind of design or come to a conclusion together with the authorities what is the best approach for a particular molecule.
So, good afternoon, and my question is to Christian. So, tell me please, from what we know, March 21st, 2013, in the state of Virginia, they signed a law that defines the conditions specifically for a pharmacist, for a prescriber, to replace the original product with an interchangeable biosimilar product. So my question to you would be, are you experiencing to spread this law in other states? And does that mean that in the United States there's no presumption of non-interchangeability for original product?
Right, so the question is about substitution practices at the pharmacy level in the United States. So the Pharmacy Practice Acts in each of the 50 U.S. states govern substitution policies at the pharmacy level. So even though there is a federal standard for interchangeability of biological products and then an FDA determination of interchangeability of a biological product, there then needs to be an amendment to each of the Pharmacy Practice Acts in each of the 50 states to permit the substitution of biologics. So it is recognized in the U.S. that the existing generic substitution policies do not apply to the substitution of biological products, and we will need to systematically go through and amend those laws to permit the substitution of biosimilars. some of the things that the states are considering is, one, a distinct provision in the law that requires that the FDA has designated the product as an interchangeable biological product. And second, that the physician has the right to prohibit the substitution of the biological product by the pharmacist. There are some other provisions being considered in the states, including informing the patient of the substitution prior to the substitution, consulting with the physician prior to substitution of the product, and also adequate record keeping so that you can go back and track exactly what product was dispensed at the pharmacy.
Thomas Schweidmüller-Roche. Actually, it's a question to Thomas. I was a bit surprised to hear on the one thing you were, on the one end, you were showing the slide that, well, patients are suffering all over the place around access. On the other end, you were saying you have difficulties to recruit patients in studies, because you were mentioning that this quite often is an issue in the biosimilar development. So I'm wondering, how does this go together? So in principle, if the one is correct, there shouldn't be any issue in recruiting patients.
I mean, it's all a matter of, it's all relative. Like, of course, the business would like to be fast and recruit as fast as possible, but then we also have to follow GCP, of course. We, of course, respect all the international rules for clinical trials, and so we wouldn't recruit in countries where there has been cases of like bad publicity, of patients not going through the proper informed consent procedure. So we're probably slowing ourselves down a little bit by having this highly ethical approach to our patient recruitment. But then I didn't say we have problems. I said it is of course it can be a bottleneck, or it can be the rate limiting factor. How quickly can you recruit? And for some trials, it may take 12 months or 18 months until you have recruited, let's say 500 patients, just to give an example.
Add something here. We obviously come across of issues with the enrollment of patients in clinical trials with biosimilars, and across the board there are issues. For example, with rituximab, when comparability in lymphoma or oncology indication has been required, certainly many companies experience difficulties in finding sufficient number of patients and enrolling equivalent studies in such indication. And when number of biosimilars are developed at the same time by great number of companies, and I actually quoting specifically a number we heard from Data Monitor, 40 companies develop biosimilar rituximab simultaneously. It means that we simply run out, in some cases, some of these niche indications. But apart from that, there are a lot of clinical issues with trying to identify, consent those patients. And obviously, it's not just simply any patient can go with, for example, lymphoma in such trial. The inclusion-exclusion criteria for biosimilarity trials are much more narrow often than just for any oncology trial which was developed for reference products. So there are lots of issues which beyond just this discussion, which results in the trial issues. And actually, there is data which clearly demonstrate how this translates in delays in some of these trials. And actually, there are specific discussions in press that Samsung and Teva actually halted some of the developments in rituximab primarily for the reasons of recruitment.
Elena Popov from EPM. So I have a comment specifically with respect to the questions and answers, and I think that it's a bit of a confusion now with regards to our colleagues when they ask about the interchangeability. But in the Russian Federation specifically, the experience of interchangeability is pretty much automatic. So we do it automatically. It's called automatic substitution. It's part of the existing framework of government guarantees, and it's fully in compliance with the current federal law. In other jurisdictions, we might speak about the different idea behind interchangeability and the so-called automatic substitution, different levels. So we not always mean the same thing, and the question doesn't match the answer very often. So let's be very careful about it. Let's go back to it tomorrow, and there will be a dedicated session for interchangeability, so we can talk about it later. Thank you. Absolutely, absolutely. We're going to have a discussion on pharmacovigilance and safety. So all these issues of pharmacovigilance and interchangeability will be one of the key topics. By the way, we'll have Niels Vermeer. So he's a very experienced person in this regard. He was working in the pharmacovigilance working group at the European Union, and he knows everything there is to know about the interchangeability. So will there be any other questions specifically on the topic that we've discussed today? I think that the second session was a success. We've heard from multiple viewpoints, and we've heard from US, Europe, we've heard from Russia. So I think this information is indeed very useful, and of course we need to digest that, to discuss it, sleep on it, but I think that it was a very useful discussion indeed, and we touched upon all the necessary issues, and I believe it's a very good foundation for discussing further issues, harmonization requirements towards biosimilars. So, and since we are getting slightly late, so, and I'd just like to wrap up the session. Thank you for your patience, and I would like to welcome you here tomorrow for the next day, and we'll continue tomorrow. Thank you very much.